I've had the privilege of hosting OMG OMx for over a year now, and I'm always surprised when I still learn things. I hope that you'll tune in and learn more from this episode. Hi, Chris. I'm really excited to welcome you as a guest on Booker's OMG OMx podcast series. As we often start, I was hoping you could tell us your personal science story and take us through your journey.
Okay. Well, first, let me say thank you very much for having me here, Kate. I'm very much looking forward to this. So I have a bit of a strange voyage through science because I have essentially been with one organization pretty much my entire professional career, starting all the way back in the beginning when I was in high school. So that's where I'll start telling you my story. So I have an older brother, Doug, who while I was moving into high school in say, freshman year of high school, he was already doing his PhD in biochemistry at Northwestern. And so I kind of saw that there was this thing, a career in science that was out there without having to learn that organically. So I think it gave me a bit of a head start there. But we were both inspired by the same high school teacher, a man named Dave Ryan, who, even though it was 10 years apart, got us really excited about science. And it turned out that Dave knew Don Combe at New England Biolabs, and he was able to get me an internship when I was 18 years old, my senior year in high school, through Project SCORE through my local high school. And it's interesting, but that simple thing changed the arc of my entire career, if not my entire life. So I worked at NEB, that internship, and then summers and whenever I could, all the way through college when I was at Colby College in Maine. And then not knowing if I wanted to go directly into graduate school at the end of college, I went on board with NEB as an entry-level scientist for a few years while I figured it out. And again, another one of those pivotal moments was there was a lot going on at the company at that point in time. So this is now the early 90s. And Rich Roberts had just moved from Cold Spring Harbor. Rich was our CSO at New England Biolabs. He just moved from Cold Spring Harbor to NEB. During that period of time, he was recognized for the Nobel Prize and for work that he had previously done at Cold Spring Harbor. And he was preaching as he came over to NEB that we needed to move into other areas outside of restriction enzymes. And one of the areas that he was preaching was modifications of proteins. And so I remember he wrote this one really nice publication that kind of described phosphorylation of proteins and glycosylation. And I got very interested in that. And so I ended up working for him during those years and a PI named Ellen Guthrie. And she was one of the first to raise her hand and say we wanted to clone exoglycosidases. So we did that, and I got exposed to carbohydrates for the first time. And again, that changed the arc of my career. I knew I wanted to go to graduate school, and I knew I wanted it to be in glycobiology. It was also right around that time that Genzyme had been approved for one of the first biologic drugs that included, as part of their processing, modification of the carbohydrate using exoglycosidases. And so it was really kind of an exciting time because I could see that there was this area that I was interested in and the direct application of that in pharmaceutical science. And so my long-term interest in enzymes that act upon carbohydrates was born from this period. So I eventually left NEB, got my doctorate at the University of Illinois, and then later found my way back to NEB, where I came on this time as a principal investigator and started a lab working on glycoprotein expression in eukaryotic microbes in yeasts. And then from there, became a staff scientist and ultimately a scientific director, where I ran one of the research divisions focused on protein modifications glycosylation, and protein expression, bringing all of those lessons together, and ran that for 18 years. And so back in September of this past year, I, after 27 years at NEB, retired from NEB. So I proceeded after that to do a bunch of fishing, a bunch of hanging out with family and friends, and decompressing. And but I'm happy to report I'm jumping back into the game and coming to a small nonprofit organization, Gloucester Marine Genomics Institute in Gloucester, Mass. And I will be heading up their protein discovery and innovation program.
So you've done so many different things. You were with NEB for a very long time. You got to watch the company evolve. You got to take inspiration from scientific advancements. But tell me how you found your source of innovation and how you found the desire to stay in the workplace even after retiring.
So in terms of finding the inspiration for innovation at NEB, I really pushed that back to NEB as an organization. They did a remarkable job, specifically Don Combe, in how he set up the company originally with a research-centric focus. But it was just an amazing climate in which to do research. You were listening to people that were doing research in all different topics throughout biochemistry and molecular biology and enzymology. And then you were going to conferences and you were interacting with academics in the field. You were interacting with other companies in the field. You were constantly getting information on what the most interesting problems were out there. So finding ideas to work on, that was easy. You had a climate of collaboration and innovation across 30 groups that were doing this. And then I would say we also had the freedom to chase our own ideas. And this was really why I stayed with one organization my whole career. I was privately funded to chase my ideas. Who wouldn't want that? That's a gift. And so, when you don't have to do fundraising and write grants and can do cutting edge science and work with academics and publish and everything, that's pretty special. And so it's why you find a lot of people from NEB have been career-long researchers there.
So then what about going back into the workforce? I'm glad you got your time to do fishing. That's important. But, what was part of your desire to not stop yet?
Yeah, I think it's an active mind. I don't know. I was pretty tired. I was pretty burnt out. But it's funny. You do some fishing trips. You do some of the things that you love to do. And then as you find relaxation, that mind takes over again, right? And so I'm finding myself missing some of the science that I was working on. I found myself watching a lot of documentaries, diving back into the literature and reading papers that I've been meaning to read for years, but just the job never lets you get to reading a long list of papers. And so I got super energized by that. And as I became more rested and as the opportunity came forth, it became clear to see that I wasn't done in science yet.
As a mid-career person, that was really nice to hear that, that things can still evolve as it goes on.
Absolutely.
So maybe let's stick with that science theme. You started your research career in glycobiology. You persisted with that for a very long time. Can you tell us a little bit more about the field and the practical uses that you already kind of hinted at?
Absolutely. Well, I think the first thing that we have to set here is a baseline about glycobiology, because it is an absolutely massive field. If you think about where sugars lie, it's in every part of cell biology, from nucleic acids to decorations on proteins to decorations on lipids. So the biology that the glycome touches is absolutely massive because it's everywhere. So now my slice of glycobiology has always been slanted a little bit more towards the applied. I was looking at enzymes that do physical things, act upon glycans, take them apart in very specific ways, build them out in very specific ways. Obviously, if you're working on enzymes like that, you find yourself working adjacent to a lot of analytical techniques. And so my road through glycobiology has always been close to the analytical side of glycobiology. There, I think that there is still tremendous potential and importance going forward in the field. Technologies have wildly improved over the last 20 years, let's say, analytical technologies. And I think we're now just arriving at the point where we can deal with some of the complexity that we have in the glycome, because technology has finally caught up with it.
So tell me about how mass spectrometry has influenced this field, especially when you started. What was the first type of mass spec experiment that you did, and what was the last thing that you did in your time at NEB?
Well, it's interesting because early in my career, mass spec was one of those things I never, ever saw myself doing. I was heavily into biochemistry and genetics, and mass spec was something that specialized labs did because they had the equipment and they had the know-how, and it was a little scary for me to look at. And I would put that in the 1990s type time frame. Back then, you had people that were pretty much characterizing individual glycoproteins, and they tended to use different types of mass spec depending upon the lab that was doing the work. Different labs had their different flavors. I think over time, there was a little bit more coalescence around the types of analyses that were being done in mass spec. So my first foray into any kind of use of mass spec in my program was actually in proteomics, not glycomics, and that was in the 2000s, 2010-ish, where we were starting to identify proteomes that had been secreted from yeast cells, and trying to understand the repertoire of glycoproteins that were normally produced, and so that we could understand better what was contaminating our proteins when we were trying to express, for example, heterologous proteins for a commercial purpose. From there, we got into glycosidases, more glycosidases, and liberated glycan analyses, and that's where we started to get into the LCMS type workflows. And so that's when my lab really started to do their first glycan-based mass spec approaches. And again, we tiptoed into it. I don't want to purport that we were experts in this. We tiptoed into it through collaborations with people like Pauline Rudd and National Institutes of Bioprocess Research and Training over in Dublin, Ireland. And then over time, we started doing more of it ourselves, but we had a lot of help getting there. And then as of late, as I was wrapping up my time at NEB, we were starting to venture more into, instead of liberated glycan analysis from individual proteins, we were starting to get into true glycomics analyses of entire glycomes all at once. And I was very interested in the extracellular matrix and the wild complexity you have of structures there, and using mass spec imaging to try to visualize that particular environment, and then enzymes to manipulate that type of environment.
So you did a lot with the bulk view of the glycome. And do you think that imaging has a role in that, or is it going to continue to be for very targeted avenues and workflows overall?
No, I'm still very bullish about imaging as being key to our understanding. If you think of where we've come as a field, we've gone from, in just a couple decades, being able to only manage glycans from one protein to then, mixtures of proteins, but not anything wildly crazy yet, to now looking at glycans in situ in a highly complex environment like the cell surface. So now starting to understand how they're sitting there, how they're arranged, what are they in proximity to, will affect so many things, I think, from diagnostic strategies to even therapeutic strategies, by designing ways to, perhaps bore a hole so that a monoclonal antibody can have greater access to its target, a receptor that you're trying to stimulate or something like that. A kind of three-dimensional view, I think, is going to give us a lot of information that will help the field translate.
So you mentioned a time point. Glycobiology has really only been around for a few decades. I remember one of the early science articles where people finally realized we don't need to cut all the glycans off of everything in order to do our analyses. So when do you think that table turned? When did glycobiology start being taken seriously and recognized?
I think you had pioneers that were looking at enzymes that acted upon glycans back all the way into the 60s and 70s. And you ask different people this question that have been around for a long time, they might view it slightly differently. But for me, again, it was that genzyme biologic drug where it wasn't just about the protein anymore. It was about the protein and the precise composition of the glycan that was appended to the protein that gave it efficacy. And then you have soon thereafter, say within a decade, growing throughout that 10-year span, the emergence of the first monoclonal antibody drugs, and then ultimately an explosion of monoclonal antibodies in the clinical pipeline. Monoclonal antibodies each possess an N-linked glycan in their Fc domain. And so, the early days of monoclonals, it was about understanding the composition of the glycan because you needed to be able to convince the FDA that you were manufacturing the same molecule over and over again. But over time, people started developing anti-cancer, for example, anti-cancer monoclonals. And that's a situation where you need the antibody to bind on an epitope on the cell surface, but you need to recruit an effector cell that's going to come in and actually do the killing. And that's all mediated by the composition of the glycosylation on the Fc domain. So, you start to see the emergence of the importance of this field that tracks very closely with the more that we know about biologic drug development. And I think biologic drug development, and I'm hoping a clinical development, glycans in the clinic in the form of diagnostics, I see that as likely driving the sector for many years to come.
Is that where you think the ultimate success is going to come from in glycobiology?
I think so. I think you can certainly make arguments about biofuels and all the other places where carbohydrates end up. I tend to look at the world as carbohydrates as they pertain to proteins, and certainly within the protein space, I think translation of our field into diagnostics and is going to be a huge hallmark of success. Upwards of 4% of proteins in the human proteome are in some way predicted to be involved in glycosylation. And maybe not directly involved in manipulating, but maybe it's pathways that are feeding into glycosylation. But that's a lot of genetic resource, a lot of genes that are applied to that particular molecule. So as cells change, whether it's programmed physiological changes in our body, like adolescence or menopause or pregnancy, or lifestyle changes, you're a smoker or you're obese, which affects gene expression patterns, or diseases like cancer that makes changes in gene expression patterns. As you change gene expression patterns, you're going to hit some of these pathways, and you're going to change the composition of the glycome. And so in that sense, the cell itself is a bit of a glycomic biosensor, if you will, as to what's going on inside the cell. It's hard to look away from the diagnostic potential of a system like that. So I think that's going to be a major hallmark for success in the future is understanding that so that we can actually action that.
So let me ask you one more maybe science question. Now that you're at an institute that's focused on protein discovery from the focus of the genes, how are you going to incorporate that in, and what do you hope to achieve in your new role?
So, well, first of all, thank you for asking that too, because that's a big topic here right now. But I think the folks at GMGI have recognized that as a genomics-focused organization, there's much that they can do. Never before has science had greater access to genomes than it does right now with deep sequencing approaches. but that's only one piece of the puzzle. It's really what the genes encode that give a greater understanding of biology. And so, if you take E. coli, for example, E. coli, I think the genome for E. coli was first released in, I want to say 96 or 97. So a long time ago. And yet 35% of the genes in E. coli are still not functionally defined. So if you then go to a space like the ocean and start to look at microbes that haven't even been discovered yet, from ecological niche that are poorly understood, and we don't even know the composition of what is there, and you start asking, what's the genetic composition, and then what is the function of proteins in those environments, your potential for discovery is utterly massive. And if you take that, what you can find and start to apply it to really good problems, you have a real serious opportunity to help affect analytics, help affect human health, help discover new therapeutics, and so forth. So I think they go hand in hand. And so my job coming up is going to be to assemble a world-class screening and protein characterization program that will start to mine this resource, so that we can start to turn that back on some of the old problems.
Well, it sounds like you're going to return to a highly science-focused life, and fishing might not be enough to calm you down. So, do you have any other hobbies that help you to kind of decompress, take you away from the grind?
I definitely do. Look, fishing is here to stay. And, if you look out the window here, I think there's going to be a fishing rod in my office eventually, so that I can rush out if I see some action going on. But yeah, no, I have a highly technical hobby that I've been doing since I was in college. So, 30 plus years ago here, which is amateur radio, ham radio. And it's held my attention for a very long time.
Tell me a little bit more about how ham radio technology has evolved. Because when I think about this, I think about an old analog system and you're tweaking the dials, but
Yes.
I know there are connections to modern technology.
Those old black and white photographs of,
Yeah.
of the radios with the big dials and somebody's grandfather sitting there. And no, I think that's how a lot of people used to get involved in it. When I first started in the hobby, it was interesting because there was still a ton of emphasis on Morse code. Those that were really hardcore in the hobby really wanted to learn and know Morse code. But once you did, you would get on the radio and you would hear the guys that were really, really good at it. And they were all former ship operators from the World War II era. And it was really an amazing chapter. But over time, we started to lose that generation, of course, and also technologies evolved. And I think the difference now from just, say, 20 years ago is the amount to which we are integrating computers into radio. And so a lot of radios, in fact, the radio that I use is actually something called a software-defined radio, where the interface to the radio is 100% on my desktop here. I control every aspect of it, and all I'm receiving from the radio is audio or microphone going out to the radio, but it's essentially a computer. But then there is, the advantages that computer transmissions are giving us, and this has certainly been known in space for a long time, but digital telemetry is something that can go farther distances. And so the field is doing less and less talking, less and less Morse code, and more communication through digital modes.
Chris, it's been so great chatting with you today, and I just wanted to thank you once again for coming on to the series.
Well, Kate, thank you again for having me. I thought it would be fun to say goodbye to you the way that ham radio operators say goodbye to each other on the air when we're doing Morse code. And so let me send this little bit of code to you.
Did that go as you guys expected? Because I really didn't expect name dropping from proteomics, glycomics, the lipid sphere, pharma, and imaging all in one episode. I hope that you found value here because I really enjoy bringing these to you.